My name is Hajra Yunus, and today we are going to discuss about prokaryotic DNA replication. In the last lecture, we discussed about what are the requirements of DNA replication. So in this lecture, we will discuss about uh, prokaryotic DNA replication. So in this lecture, we will cover the topics such as initiator proteins in case of prokaryotes, helicase loader and helicase loading, DNA polymerase, primase, process of polymerization, and termination. So firstly, we will start from initiator proteins. Okay, so as you can see in this diagram, so this is the structure of origin of replication in case of bacteria. Okay, that is ORIC. Origin of replication in case of bacteria. As you know that in case of bacteria, origin of replication is almost 245 base pair long. Okay, and initiator protein in case of prokaryotes is the DNA. Okay, so DNA is the initiator protein that helps in the unwinding of DNA strand at the point of origin of replication. As you can see in this diagram, in case of prokaryotes, there are 13 mer sites are present. Okay, they are the 13 mer repeats, 1, 2 and 3. Okay, so there are 13 mer repeats and there are 9 mer repeats are also present. So there is a one point that I need to tell you, that in some books it has written that there are 1, 2, 3, as this diagram shows that there are 4 9-mer repeats, but in some books it has written that there are 5 9-mer repeats, but in most of the books there are 4 9-mer repeats. Okay, so we will follow that there are 3 13-mer repeats and there are 4 9-mer repeats. Okay, so in case of origin of replication, we are just talking about origin of replication in case of bacteria. So this is the structure of origin replication in case of bacteria. And one more thing, origin replication is AT rich region. Adenine and thymine are rich in number compared to the guanine and cytosine, and because of high AT rich region, melting temperature is low, and because of the low melting temperature, it is easy for initiator protein to unwind the DNA strand. So this is the initiator protein. Now in the next slide, we will look at how the initiator protein works. So as you can see in this diagram, okay, as you can see in this diagram, here you can see that these are the initiator proteins. Okay, these all are the only not only one initiator protein, that is the DNA in case of the prokaryotes attaches to the DNA strand, but there are many initiator proteins. So firstly, DNA is the initiator protein, and that initiator protein firstly this initiator protein starts attaching to the 9-mer site. So these are the 9-mer site. Okay, so these are the 9-mer site. Initiator protein attaches to the 9-mer site, and here the one initiator protein attach, then another initiator protein attach, then another attach, and another attach. Okay, so in that case, the initiator protein, number of initiator proteins attaches to the DNA strand, and once the initiator protein reaches to the, as you can see here, let me change the color. So these are the, oh, another color, let me change another color. Yes, as you can see here, these are the 13-mer sites. These are the 13-mer sites. So firstly, how the initiator protein helps in the unwinding of DNA strand. Firstly, the initiator protein attaches to the 9-mer site, and once the initiator protein reaches to the 13-mer site, then unwinding of the DNA strand takes place. So this is very important thing. The important thing is that the unwinding of the DNA strand does not start on the 9-mer side. Okay, it starts on the 13-mer side. Firstly, the initiator protein attaches to the 9-mer side, and once the initiator proteins reach to the 13-mer side, then the unwinding of the DNA strand takes place. As you can see in this step, as you can see on this step, in this step here, once the initiator proteins reach to the 13-mer side, here you can see that unwinding of the DNA strand takes place, or here the DNA strand has been unwinded. So this is very important step, and in most of the competitive exam this question comes that, or this question is asked that on either the unwinding of the DNA strand starts on the 9-mer side or 13-mer side. So the exact answer is that the initiator protein, that is the DNA, attaches to the 9-mer side, and when the DNA reaches to the 13-mer side, then on 13-mer side unwinding of the DNA strand takes place. Okay, and one more thing that is very important, this is the ATP dependent process. Okay, so unwinding of the DNA strand by initiator protein is the ATP dependent process. In that case, ATP is converted into ADP. As you can see, ATP is converted into ATP. Now there is the unwinding of the DNA strand, two strands get separated, and now in the next step there will be the loading of helicase. So one important thing is that helicase cannot load on the double strand. For the helicase, single strand is required. So all these things are going on, or all these things are taking place for the loading of helicase. If there is no single strand, there will be no loading of helicase. If there is double strand, then of course there is no loading of helicase. As long as there is a single strand, then helicase is loaded by helicase loader. So in the next slide we will discuss about what is the helicase loader, and how helicase is loaded on the DNA strand with the help of helicase loader. I hope in this diagram is you can understand this diagram, and you can get this diagram, because this diagram looks difficult but it's pretty simple. So the main thing in this diagram I want to tell you that the initiator protein helps in the unwinding of DNA strand. So when it comes to the origin of replication is 245 base pair long, and it has 13-mer repeats, 3 13-mer repeats, and 4 9-mer repeats. Okay, and firstly the initiator protein, that is the DNA, attaches to the 9-mer side, and once the DNA or initiator protein reaches to the 13-mer side, at that time unwinding of DNA strand takes place on the 13-mer side. So, and this is the ATP dependent process, energy is required. So in this slide we will discuss about the helicase loader, and how helicase loader is helpful in the loading of helicase enzyme. Okay, so you know that the helicase is very important. It is the important enzyme, or it is important for the unwinding of DNA strand. If there is no helicase, or helicase does not sorry, if helicase does not perform its function properly, then there will be no unwinding of DNA strand. If there is no unwinding of DNA strand, so in that case there will be no DNA replication, or process of DNA replication will be affected. So now you can see here the helicase loader. In case of prokaryotes, helicase loader is DNA-C and helicase is DNA-B. Whereas when you compare helicase loader of prokaryotes and eukaryotes, you will know about that. Like I discussed in the last lecture, that there is a difference between helicase loader in prokaryotes and helicase loader in eukaryotes. Although the function is similar, but in case of prokaryotes there is only one helicase loader, whereas in case of eukaryotes there are two helicase loader. That would be CDC6 and CGT1. So okay, and how they both function, we will discuss about when we will discuss about eukaryotic DNA replication. Okay, now you can see in this diagram the DNA, double strand DNA has been separated. Now they are two single strand. Now why the unwinding of the DNA strand is required by the initiator protein? Because the helicase loader or helicase needs the single strand. As long as there is a single strand, then the helicase will be uploaded by helicase loader. If there is no single strand, in that case there will be no place for helicase to be on the DNA strand. So single strand is required for the loading of helicase. As you can see in this diagram, here the helicase loader is the DNA C and the helicase is the DNA B. So DNA B is the helicase and the loading of helicase is required. So you can assume that why the helicase loader is required. The helicase loader is required because the helicase does not know the location where it will be loaded or where it should be on the DNA strand. So in that case, the DNA C, that is the helicase loader, that guides helicase on the DNA strand. Okay, so as you can see in this step, the helicase has been loaded on the DNA strand on the both of the single strand. So this is one single strand and this is another single strand. So the DNA helicase has been loaded on the DNA strand with the help of the helicase loader. So the function of helicase loader is only to load helicase on the DNA strand. After performing its function, as you can see in the next step, that helicase loader will leave the DNA strand. As you can see here, the helicase loader has left the DNA strand. After leaving the DNA strand, the next step will performed by is the primer formation, that is done by DNA G. Okay, in case of the prokaryotes, the primer formation or the primase activity is performed by DNA G, and after that the process of the polymerization will be carried out. So this is about the helicase loader. Okay, so in case of prokaryotes, the helicase loader is DNA C and the helicase is DNA B, and for loading of this one helicase DNA B, DNA C is required, and after performing its function, DNA C will leave the DNA strand and the other processes will be carried out by their respective enzymes. So in this diagram you can see that we will discuss about clamp, let me change marker, clamp loader and DNA polymerase. Okay, as we discussed in the last lecture that there are different types of polymerases in case of prokaryotes. So let me repeat one more time. In case of prokaryotes, there are 5 different types of polymerase. That is 1, then 2, polymerase 2, polymerase 3, polymerase 4, and polymerase 5. There are 5 different types of polymerases in case of prokaryotes. The polymerase 1 has the polymerization activity and exonuclease activity also. Okay, so polymerization activity and exonuclease activity. When we say that exonuclease activity, it means that the exonuclease activity is from 3' to 5' direction, and polymerization activity is from 5' to 3' direction. Okay, so and polymerase 2 is responsible for the base repair also. Okay, and the polymerase 3 is the basic is the main polymerase in case of the DNA replication in case of prokaryotes, and this diagram shows the structure or holoenzyme of DNA polymerase 3. Polymerase 4 and 5, they both are responsible and they both work under the difficult situation. When there is a difficult situation and process of polymerization is not performed by DNA polymerase 3, in that case DNA polymerase 4 and 5 will work together. But there is one important thing is that they have the polymerization activity but they do not have exonuclease activity. Okay, if there is no exonuclease activity and there is only polymerization activity, so in that case, if there is a uh, mismatch base in the DNA strand, so in that case, if the polymerization is performed by polymerase 4 and 5, in that case they will not go back and will not repair the mismatch base pair. So in that case, so it will be and leading to a mutation or the DNA damage. But polymerase 1 and 3, and even they have the polymerization activity and the exonuclease activity. So if there is a problem, they can go back and they can repair the damage, or they can repair the mismatch base. So when we talk about the clamp loader, beta clamp, so in case of prokaryotes, there is a clamp is present. So this is the sliding clamp. Okay, so sliding clamp is called the beta clamp in case of prokaryotes, and in case of eukaryotes it is called something else. Okay, I will tell you the name when we will talk about the prokaryotic DNA replication. Now next thing is required is the clamp loader. As you can see in this diagram here, the sliding clamp is present and the clamp loader is present. Okay, and the third thing is the polymerase, DNA polymerase, and here the DNA polymerase is present. Okay, so as you can see in this diagram, there is a sliding clamp, and the sliding clamp is responsible to hold the DNA strand. Okay, now because the sliding clamp loader will provide the space for the attachment of polymerase 3. Okay, but for the attachment to the DNA strand, for that the sliding clamp is required. So this is the sliding clamp. Firstly, the sliding clamp attaches to the sliding clamp loader, and now this sliding clamp will attach to the DNA strand. Because this is very important, if there is no sliding clamp, so DNA polymerase will not perform its function and will dissociate after some time. But the DNA polymerase continues its activity because there is a sliding clamp that can hold the DNA strand. So as you can see in this diagram, sliding clamp, or that is the beta clamp in case of prokaryote, is holding the DNA strand, and DNA polymerase, this is the DNA polymerase, and DNA polymerase is performing its function. So as you can see in this diagram, there are three arms. One arm, this is two arm, and another is the three arm. So there are three arms are present. So why there are three arms? So these three arms are involved in the process of polymerization. So out of three arms, two arms are flexible and are more flexible than the another one arm, because two arms are responsible for the polymerization or for to perform the DNA replication on lagging strand, and the only one arm is responsible for polymerization on leading strand. As you know that the leading strand is continuous strand. If there is a continuous strand, or leading strand is a continuous strand, and that goes smoothly or that goes continuously, so only one strand or one DNA polymerase is enough for polymerization. Whereas in case of lagging strand, on lagging strand the replication takes place in fragments, or there are Okazaki fragments are present. So in that case there are two polymerases work together. If the two polymerases work together, so they will change. Firstly, they will work on the one place, then they will change the place and they start working on another place. So in that case, the process of DNA replication is completed. So another thing is very important, that is the clamp loading ATP dependent process. So loading of the sliding clamp requires ATP. So this is the ATP dependent process. By using the energy, or by at the expenditure of energy, or ATP is converted into ADP, and then in that case sliding clamp, or that is the beta clamp, attaches to the sliding clamp loader. Next is the primase activity and polymerization. Okay, primase activity and polymerization. So this first diagram is about the primase activity. What is primase activity? Primase, as you can see, that primase means the primer formation. It is responsible for primer formation. So here the primer is this blue one. As you can see, this blue one is the primer, and this is the helicase, and these green are the single stranded binding protein, because when the helicase enzyme is performing its function and now the DNA strand is single stranded, so the DNA strand may try to form bond again. So to prevent the rewinding of the DNA strand, single stranding binding proteins are attached, and they will not let the DNA strand to rebind again. So single stranding binding proteins are very important. They do not recognize they are not nucleotide specific. Their function is just to attach on the DNA strand after nuclease enzyme performs its function or complete its function. And the second thing is that the bond formation between the single stranded binding protein and the DNA strand is the electrostatic interaction, that is the weak interaction. So after, when the DNA polymerase starts performing its activity, then the single stranded binding protein will leave the DNA strand easily. As you can see here, the blue color is the primase. So this primase is responsible for the primer. So here you can see the RNA primer leading strand and RNA primer on lagging strand. So there are two helicases, one helicase is on leading strand, one helicase is on lagging strand. So in the same way, the one primase will be on or will work on the leading strand, and one primase will work on the lagging strand. So in vitro case, here the RNA primer is formed. So whether the DNA primer or RNA primer are formed, so the polymerization process is not affected, or DNA polymerase can perform its function smoothly. But in vivo, like in our in vivo, RNA primer is formed, but in vitro we use DNA primer, like in case of the PCR. So whether the DNA primer and RNA primer, it doesn't matter. The basic thing is that the DNA polymerase needs something, or you can say DNA polymerase needs 3' hydroxyl group. As long as there is a 3' or hydroxyl group, so 3' hydroxyl group is required by DNA polymerase 3. If there is a 3' hydroxyl group, so in that case DNA polymerase 3 can perform the process of polymerization. So this is very important. Sorry. Now in this diagram you can see that now there is a sliding clamp, there is a sliding clamp loader, there are the 3 arms, and they are working together. As you can see, this is the leading strand, okay, and here this is the lagging strand. So on lagging strand 1 or 2 DNA polymerases are working, and the one DNA polymerase is working on the leading strand. As I told you in the last slide that for the leading strand only one DNA polymerase is enough, because the leading strand is a continuous strand. There is no breakage, there is no halt. So if there is only continuous strand, so in that case only one DNA polymerase 3 is enough. So DNA polymerase 3 is performing its function continuously, whereas in case of the lagging strand, because you know that the polymerization takes place from 5' to 3' direction, so here the process is continuous, or process keeps going. So everything is fine, but here the process is carried out in fragments. Like here, firstly the primer will attach here, and the process of polymerization, here the 5' hydroxyl group, and here the 3' hydroxyl group is available. So here the 3' OH group is available, so the DNA polymerase starts its activity here. So the polymerization will take place to this direction, and after that here if there is an unwinding of DNA strand, here the another primer will be attached by the primase, that is the RNA primer, and here the process of polymerization takes place. So in that case, on the lagging strand, two DNA polymerase 3 are working together. One will work on the one point, another will work on the another point, and after completing their task on that point, now the helicase is performing its function simultaneously. Helicase is unwinding the DNA strand. Now there is more part of the unwinding of the DNA strand, or there is more part that has been unwinded by helicase enzyme. In that case, the DNA polymerase will go to that place and start process of polymerization over there. So in that case, by changing their position on the lagging strand, the process of DNA polymerization will be carried out by DNA polymerase 3. Okay, so firstly, the first step was the initiator protein. The next step was carried out by helicase loader to load helicase, and after loading helicase, helicase loader leaves the DNA strand. Next step was the uh, clamp loading, and that is the DNA polymerase. Another step was the primase activity, and after the primase activity, the process of polymerization. Now the 3' hydroxyl group is available, and after that, now the DNA polymerase will start its activity. Now there is another question, and that is very important question. Why during the process of polymerization DNTPs are used, not RNTPs? Means deoxynucleotide triphosphate are used, whereas not RNTP, that is no ribonucleoside triphosphates are used. So the because the question comes on our mind that because in our system or in our body RNTPs are more in number or compared to DNTPs. So why during the process of polymerization DNTPs are used, not RNTPs? So the answer is that because of the functional groups. Sorry, functional group, because of the functional group. As you, this structure is of DNTP and this structure is of RNTP. Okay, as you can see. So this is the DNA polymerase, the green in color, and here the DNA polymerase also has the discriminator amino acid. Okay, and that discriminator amino acid is the tyrosine. Okay, so now as you can see, in case of DNTP there is only one OH group is present, whereas in case of RNTP 2 OH group is present. 2H group are present. And now because you can see here, if we talk about if there is a DNTPs are used, because of the DNTPs you can see here the OH group of the, here the adenine and thymine and adenine and thymine. Okay, now if DNTP is used, in that case as you can see here the discriminator amino acid that is present, that is the tyrosine, and that tyrosine will recognize or can recognize the DNTP, and this tyrosine can fit OH group of the DNTP here in this bulging. Okay, so after the discrimination, that is the tyrosine can recognize the DNTP, and after recognizing DNTP, tyrosine will fit the or can fit the hydroxyl group of DNTP in this bulging, and after fitting here, as you can see here, their OH group is present. So to make form between the two nucleotides, this OH group will attack on the alpha phosphate of the nucleotide, and after attacking on the alpha phosphate of the nucleotide, the bond formation will carried out. Okay, so everything is correct. If there is everything is correct, if DNTP is used by the DNA polymerase, in that case firstly the discriminator amino acid of the DNA polymerase, that is tyrosine, will recognize the DNTP, and after recognizing DNTP, this tyrosine will fit OH group of the DNTP in the bulging, and after adjusting or after fitting in the bulging, here the nucleotide, OH group of the one nucleotide will attack on the alpha phosphate, and then the bond formation will take place like this. Okay. Now in case if DNTP were not present and RNTP was used, in that case you can see that here the two hydroxyl group are present, one hydroxyl group, another hydroxyl group. If there are two hydroxyl groups, so tyrosine can recognize only DNTP, tyrosine cannot recognize RNTPs. If tyrosine cannot recognize RNTPs, so in that case there will be no fitting of hydroxyl group in this bulging. If there is no fitting of hydroxyl group in this bulging, so because of that, what would be the effect? That there is the distance between the hydroxyl group and alpha phosphate will increase. If the distance between hydroxyl group and alpha phosphate will increase, in that case the hydroxyl group will not attack on the alpha phosphate of the nucleotide. If there is no attacking, so in that case there will be no hydrogen, no phosphodiester bond between two nucleotides. So that's why during the process of polymerization, DNTPs are used by DNA polymerase, not RNTPs. Okay, the question is that if there is, the answer is that if their DNTPs are used, in that case the tyrosine, that is the discriminator amino acid, that can recognize that and fit the hydroxyl group in the bulging, and the process will go smoothly. But if there are two functional groups or hydroxyl group, in that case discriminator amino acid will not recognize them. If there is no recognition, there will be no fitting in of hydroxyl group in the bulging, and now because of that the distance between OH group and the phosphate group will increase. If there is no bond between OH group and the phosphate group, there is no formation of phosphodiester bond, so in that case the process is affected. That's why DNTPs are used, not RNTPs. Now, so this is very important slide, that is the polarity of helicase and DNA polymerases. Okay, as you can see here, it has written that in DNA helicase associated with DNA polymerase, so in that case helicase DNA activity increases. So this diagram shows that when, like you can see here, this is the helicase enzyme, and this helicase enzyme is associated with the DNA polymerase. So this is the DNA polymerase. Let me change color. Polymerase. So if there is an association between DNA helicase and the DNA polymerase, in that case the DNA helicase activity will be increased, and if there is an increase in the DNA helicase activity, so ultimately the process of polymerization will go smoothly. Okay, because they both are working together. So in that case, when they are working together, in that case the definitely, or it's obvious that the process of polymerization or polymerization activity will be improved. But as you can see in this diagram here, there is no association between helicase and there is no association between the DNA polymerase. In that case they are apart from each other. If they are apart from each other, so in that case the helicase activity will be reduced. As long as they are working together, if they are working together, so the process of polymerization will go smoothly. And if they are not working together and helicase is moving fast and helicase is fastly unwinding the DNA strand, and DNA polymerase is left behind, and DNA polymerase is doing its work in to polymerize the DNA strand, but still the DNA polymerase is left behind. In that case, there would be the possibility that there is coiling of DNA strand. So in case of coiling of DNA strand, so process of DNA replication is affected. So polarity of the helicase and DNA polymerases, they both are very important. If they are working together, so helicase is unwinding the DNA strand, and simultaneously because the DNA polymerase is attached to the helicase. So the helicase is or DNA polymerase is responsible for the polymerization of DNA strand. But if there were no polarity, in that case the helicase is unwinding the DNA strand fastly and DNA polymerase 3 is left behind. So in that case there would be the chance of coiling. If there would be the chance of coiling, so when the DNA polymerase and there is a coiling of DNA strand between DNA polymerase during this region, you can assume that if there is a coiling during this region, between this region, and if there is a coiling and DNA polymerase reaches to that point, and if there is a coiling, so in that case the process of DNA replication is affected. So the polarity is very important. Next is the in the end, the next thing is the termination. Okay, now the termination, the DNA polymerase or process of DNA replication has completed. Process polymerization has completed by DNA polymerase 3. So next is the termination. So as you can see here, the helicase is unwinding DNA strand and simultaneously DNA polymerase 3 is performing its function. Now how the termination will carried out or will carry out in prokaryotes. As you can see in this diagram here, the protein is present which is called the TER-TUS protein or TUS-TUT protein. When there is a TER-TUS protein, so this is the non-permissive protein. Okay, so it will if there is a, for example, if that is a permissive protein, in that case when the helicase enzyme reaches to that place and the TER-TUS protein will fall from the DNA strand, so there will be no end of DNA replication. But the TER-TUS protein is the non-permissive, so it will not leave the DNA strand. When TER-TUS protein will not leave the DNA strand, and when the helicase enzyme or the DNA polymerase reaches to that point, in that case it shows that now you need to stop the DNA replication. Okay, and after that the DNA replication is stopped, and you can say the process of the DNA replication is about to complete because two things are required more. So after the termination of the DNA strand or after the termination of DNA replication by TER-TUS protein, the next step is the removal of primers. You know that on lagging strand there is only one primer, but in case of, so this is the lagging strand, this is the leading strand. Let's write on leading. So here only one primer is present, that is 5' side and 3' side, and here the process of polymerization is carried out. Now this is another strand, this is 5', 3'. So here the replication was carried out in 3'. So here the replication was not smooth, so here the many primers are present. So this is lagging strand. Okay, now after the termination of DNA replication by TER-TUS protein, next is the removal of primer. So this primer needs to be removed. Let me change. So this primer needs to be removed, this primer needs to be removed, and this primer is also needs to be removed. Now the primer removal is carried out by polymerase 1. As you know that prokaryotes have 5 different types of polymerases, 1, 2, 3, 4, 5. So now firstly the polymerase will perform two functions, or polymerase 1, DNA polymerase 1 performs two functions. The first function is the primer removal, and the other function is the polymerization on the removed region. So what would happen that firstly DNA polymerase 1 will remove those primers, and also so you can say here DNA polymerase okay, so DNA polymerase firstly removes the primers, and after that here because this is the linear actually in prokaryotes it's a circular structure, so there is no end replication problem. So I'm just going to perform the function of polymerization here. So here the polymerization will be carried out by DNA polymerase 1, and here also polymerization process of polymerization is carried out by DNA polymerase 1. So polymerase 1, in case of prokaryotes, polymerase 1 performs two important functions. One is the primer removal, and second thing is that after removing primer, the attachment of DNPs on the removed primer site or removed region. And I made the structure linear, but actually this is not the linear structure. In case of prokaryotes, the genetic material is circular, so there is no end replication problem. So every time 3' hydroxyl group is available for the DNA polymerase 3 or DNA polymerase 1 for the polymerization process, and for the removal of primer and polymerization or removed region. Now in the end there is a small nick is present after, like you can say here in, if you can see that after the process of polymerization by polymerase 1, here the small nick is present, here is also small nick is present, so here also one small nick is present. So this small nick is repaired by the ligase enzyme. Okay, so ligase enzyme is responsible to join discontinuous strand. So the discontinuous strand is called the lagging strand, and on the lagging strand or anywhere where the small nick is present, that small nick is filled by ligase enzyme. So this is the whole process of prokaryotic DNA application. Firstly, the initiator protein is required. The initiator protein is DNA in case of prokaryotes. Then after that there is a helicase loader is required, which is DNAC in case of and then the DNAB is loaded on the DNA strand with the help of DNAC. Next step is the attachment of clamp on the DNA strand, and there is a sliding clamp, that is a beta clamp in case of prokaryotes, and then there is a sliding clamp loader, and the sliding clamp loader has 3 arms, and they are flexible, but 1 or 2 are more flexible because they are responsible for process of polymerization on lagging strand. And next step is the primase activity, and primase is responsible for, and in case of prokaryotes, the primase activity is performed by DNAG, and DNAG is responsible for primer formation, and after forming the primer, 3' hydroxyl group is available for the polymerization process by DNA polymerase 3. When the DNA replication is about to complete, when there is a tertus region, that is the termination protein. Now they tertus protein are the non-permissive, they will not leave the DNA strand. Now they will order the helicase and the DNA polymerase 3 that now you should stop your process of you should stop your activity. At that time the termination will carried out, and after that the primer will be removed by polymerase 1, and on the removed region process of polymerization is carried out by DNA polymerase 1. And if there is a join, or definitely there is a small nick is available, that small nick is filled by ligase enzyme. So I hope so you understand the whole process of DNA replication in case of prokaryotes. In the next lecture we will discuss about the process of DNA replication in case of eukaryotes. And if you have any question, you can ask question on the comment section from me. And till then, Allah Hafiz. And if you have not subscribed the channel, please subscribe the channel and hit the bell button so you can get my new video when I will upload the video. Till then, Allah Hafiz.